Dear students, this is the continuation of previous video. Cell cycle, it has two basic phases are there, which are the interphase and one more is M phase. Interphase and M phase. Interphase nothing but resting phase. On the picture we can see here, this is the M phase, mitosis phase. The interphase can be divided into three subphases, which are the gap 1 phase and synthesis phase and G2 phase. So here the cell when it's going to divide, previously we discussed about that human cell when it undergoes a division, the daughter cells will be produced within the 24 hours, whereas the yeast 90 minutes. So here this interphase, the cell gone through that, which is 95 percentage, very few, rest of 5, that cell which passes the M phase only 5 percentage, right? And here in the interphase the phase between the two successive M phases, this is the M phase, between the two successive M phases we called as interphase. It includes the cell growth and DNA synthesis. Interphase, it includes what? It's cell growth and DNA synthesis. So here what I discussed here previously, it's 95% of the duration of cell cycle which constitutes the interphase. Okay, interphase 95 percentage. And here interphase have a three phases, which are the gap 1 phase and synthesis phase and gap 2 phase. This is S phase, synthesis phase. DNA synthesis takes place here. Here they have given as a G0, that is the G quotient. Some cells which are the R muscle, which is heart muscle, these muscle cells, heart muscle cells, they should not pass to the S phase and G2 phase. There itself only they will be remain in the same phase. There is no, they are not showing any division. That's the reason G0 phase you can see in the heart muscle. Next, here is the G1 phase. In the interphase, you talk about the G1 phase. G1 phase also called the first growth phase. G1 phase, what we call first growth phase. So here, this is the interval between the mitosis and DNA replication. See, this is the G1, you know, this is the G1. This is between the mitosis and here is a DNA replication. Here it takes place. DNA replication takes place in the S phase. So between the M and S, what is there? G1 phase is there. What it? G1 phase is there. What are the main events takes place in the G1 phase? Here cell continuously grow and metabolically cells are very active in this phase, and it can prepare the machinery for the DNA replication. Means DNA replication will takes place in the next further phase, which is the N phase, S phase. For that, what are the requirements are there, they can prepare here that whole machinery. And RNA and protein synthesis also takes place in this phase, which is nothing but G1 phase. Now we'll look into the S phase, synthetic phase. In this phase, DNA replication takes place. It's a very important. DNA replication takes place. See here, DNA replication, the chromosome which is capital A and small a they have given here, it will be formed into two chromatids, sister chromatids, and here amount of DNA per cell will be doubles. Amount of DNA per cell will double in the synthesis phase. And here more thing is the chromosome number should not be increase. Chromosome number should not be increased. But DNA, for example, 2C of DNA becomes a double means 4C. But the chromosome number should be remains constant in this phase. That's very important. Chromosome number should be same, but DNA content will be double. In animal cells, replication begins in the nucleus and the centriole duplicates in the cytoplasm. Centrioles are there. See, these are the centrioles which are duplicates and it formed as they going to move into the both ends of the cell. And next, G2 phase, which is a gap 2 in the interphase, gap 2. In this phase, this is called as a second growth phase. Cell grow continuously, it's going on, and moreover the RNA and protein synthesis also continue in this phase. Cell is ready to move into the M phase. G2 phase between the what between the S and M, that is nothing but G2 phase. Means the one cell going to divide into two daughter cells. For that preparation is required. This is the one cell going to divide into two cells. For the preparation will be nothing but interphase. Here cytoplasmic content and nuclear content, everything should be equally shared. For that purpose, this much of preparation will be takes place, that is nothing but resting phase or the interphase. Now going to discuss about M phase, mitosis phase, nothing but mitosis phase. In the mitosis phase, it's actually actual cell division. It represents actual cell division. See here in this diagram they have given this is the interphase after M phase. M phase nothing but they have a prophase and metaphase, anaphase, telophase. These are the phases. These are present here, which can notice in this. Now, yeah. Now, see in human cell cycle it lasts for only about an hour. Cell cycle it lasts for only about an hour. Which is the M phase. M phase includes karyokinesis and cytokinesis. These are the very very important, karyokinesis and cytokinesis. Karyokinesis means nothing but division of nucleus. Cytokinesis means division of cytoplasm. Okay, nucleus division and followed by the cytokinesis takes place. Okay, M phase consists of the two phases, which are the karyokinesis means division of nucleus and cytokinesis nothing but division of cytoplasm. Here in this picture they have mentioned here. Here karyokinesis takes place, nothing but nucleus going to divide. Take here nucleus divides. After that the cytoplasm content, here is the furrow formed. These are going to divide into cytoplasm, equally divide. Then two daughter cells are formed from the one parental cell, right? Here is in this mitosis phase they have given, see here, nails and tip of nail and heart and muscles. What previously discussed here, some cells do not show division, which are the heart cells. Many other cells divide only occasionally to replace damaged or dead cells. Occasionally they can divide when it's damaged, tip of our nails. That area if it is cut or wounded, that will be replaced occasionally. And heart cells also, which are in the G quotient stage, I told you no, they not move for the synthesis phase. They don't have any capability to divide. And here the cells that do not divide further exist in the G1 phase and enter inactivate stage called the G quotient stage or the G0 stage. So cells remain metabolically active but do not proliferate. The cells which are metabolically active but they do not proliferate. Here mitosis is there, here in this picture this is interface G1 gap 1, and again here G0 there is a G quotient. And S phase, what happens? DNA synthesis takes place. G2 phase, after that M phase. Got it? Then here we'll go for the how it is mitosis takes place. In the mitosis, cytokinesis over and karyokinesis completed. In the in this, it is a cell division. Mitosis is about cell division occurs in the somatic cells. We know already we have two types of cells. Somatic cell is one category. Next one is the germ cells or the reproductive cells. So mitosis is called the equational division. Means as a number of chromosomes in the parent and progeny cells same. Parent cell how many chromosomes does have, that offspring which are derived from the parent they should have a same number of chromosomes. That's the reason equational division. There is no change in the chromosome number. Mitosis is generally seen in the diploid cells. You know that all somatic cells are in the 2n state, which are in the diploid state. It also occurs haploid cells. It's an exception. Lower plants and social insects, which are the honeybee. Honeybee is there, you know. A drone have a 16 chromosomes. Okay, unfertilized egg change into drone, which is the male. Okay, it involves a major recognition of all cell components, reorganization of all cell components. So here in this mitosis, first step is the karyokinesis. What is the first step? Karyokinesis. The karyokinesis of mitosis has a 4 stages: prophase, metaphase, anaphase, telophase. How to remember this in order? Here actually after this what happened na? Cytokinesis takes place. Cytokinesis. Karyokinesis after cytokinesis, you know. That's the reason PM, Prime Minister, at C means Chennai. You have to remember like that. PM at PM at Chennai means prophase, metaphase, anaphase, telophase. After that, cytokinesis takes place. Anyhow, this should be remembered in order: prophase, metaphase, anaphase, telophase. These are the subphases in the karyokinesis. After this followed by what happens? Cytokinesis takes place, which is the cytoplasmic division. Right here, prophase, what it happens here, chromosomes, how they are appeared. This is a microscope. Under microscope, this it will be visible in such way. This is the microscopic pictures. And prophase, how chromosomes are condensed. And metaphase, the chromosomes are combined and they settled into the metaphatic plate. In anaphase, they are going to pull into both ends, and telophase they completely reached. These things we can notice in the karyokinesis, which are the 4 phases. Now prophase. Prophase. Here prophase, it is the longest phase in the mitosis. Prophase is the longest phase in the mitosis. Okay, and this is the first phase of the mitosis. It follows the S and G2 phases of the interphase. See in this picture, early prophase, here is a thread-like structures there. In the S and G2 phases, DNA molecules are intertwined. Intertwined means they should be wounded like thread-like structures. What are the characters in the prophase? In the prophase, main characters are the chromosomal materials, which are the chromatin fibers, are untangled and condensed to form a mitotic chromosomes. See here, late prophase, they are condensed. They are seen in the composed. Two chromatids attached together at the centromere. One centromere is there, two chromatids are attached like this. Centrosomes begin to move. Centrosomes are there, no? Begins to move the opposite poles. Centrosomes. And centrosome radiates out microtubule called as the aster, and two asters together form as a microtubule. So the spindle fibers form a mitotic apparatus. Mitotic apparatus also formed in the prophase of the mitotic division. Cells at the end of the prophase do not show Golgi apparatus, endoplasmic reticulum, and nucleolus and nuclear envelope. These are the main characters of the prophase. Right? In the next, we will discuss the metaphase. Thank you.